Sure. So thanks for the kind invite, Ethan, for me to speak here. I'm Liang Chang. I'm a biotech investor working for Aditum Bio based in Boston. And personally, I did my PhD in cancer biology with Bill Sellers, used to be the oncology head at Novartis, at the Broad and Harvard, and working on the targeted oncology space, and have been following all the advances in the KRAS space for many, many years. And I feel like this is the most exciting data so far we have seen in the KRAS space after many years of the belief that KRAS is not druggable. And the first generation of KRAS G12C inhibitors, they are approved drugs, but the efficacy are a little bit underwhelming. So for Revolution Medicine, the drug Daraxonrasib, this drug is a molecular glue that ties on the on state of KRAS with cyclophilin A, which is a very ubiquitous protein in the cell, and that essentially displaces KRAS from binding to the downstream partners like RAF by steric occlusion. And the box in the bottom half of the slides gave some context about the data. So this is the data published on New England Journal of Medicine a few weeks ago. It is the earlier phase 1/2 trial of this drug in pancreatic cancer. They focus on the 300 mg group. Then Revolution Medicine had the press release on the phase 3 pivotal trial in second line pancreatic cancer with chemotherapy as a comparator arm, showing overall survival 13.2 months versus 6.7 months, hazard rate 0.4, which is one of the lowest hazard rate we have seen in oncology trial for a while. That data is actually going to be presented at ASCO tomorrow as one of the plenary sessions. So we will know more about that OS trial too. So today the focus is on the earlier 300 mg trial showing ORR. Next slide, please. Yeah, so this is the top line data for DARA's 300 mg. So they did, this is a dose escalation trial. They have dosed over 150 patients, and the data is mostly focusing on the 300 mg group, which they move forward with phase 3 in the pivotal trial and showing the best efficacy comparing with lower dose. This patient population is pancreatic cancer, PDAC. It is not very heavily pretreated because there's no standard care in pancreatic cancer. Standard care is pretty much just different lines of chemo. And the second line is patient experience 1 line of chemo, and third line are patients experienced with 2 line of chemo. And I have some benchmark number of historical chemotherapy data in the same patient population. So that makes analyzing pancreatic cancer data a little bit easier compared with a lot of other cancers where we deal with many different patients with different baseline, with different lines of therapy. For pancreatic cancer, simply other than chemo, nothing else works. So ORR in the second line setting for all RAS mutation is 29%, with G12 specific mutation is 35%, and duration response is 8.2 months, and median OS is 13 to 15 months. And the number drops down a little bit in the 3-line plus patient population, drops to about 20% ORR, and also less duration response and less OS. So based on this data, Revolution Medicine decided to do the phase 3 in second line pancreatic cancer. This is how they designed the pivotal trial, which we will slightly touch in a later slide. So next slide, please. Yeah, on the safety side, this is one of the concerns that people in the field have about Revolution Medicine approach, because it is not mutant specific. It inhibits all the on state of KRAS, and RAS signaling pathways play important role in the cell proliferation of a lot of different cell lineages in normal tissues. And from the safety table, it looks generally okay, and I think, better than a lot of people expected here. So the most common adverse events we see are skin and GI, which are all likely because of wild-type RAS inhibition, and it is dose-dependent. There are some grade 3 adverse events that lead to discontinuation, or dose reduction. But overall, every median patient stay on about 85% dose intensity, which is pretty good. And one adverse event that might worth noting, and that's something we need to closely watch for the ASCO presentation tomorrow, is the stomatitis or mucositis signal, because that's something that in the pancreatic cancer patient population, where the patients are already malnutritioned, it is something that might be more not as benign compared with skin and diarrhea. And also in community oncology setting, once we anticipate this drug to be approved, how would community oncologists manage stomatitis or mucositis is another question people have in mind. But overall, with the profile, especially with pancreatic cancer patient population, that should be a acceptable safety profile. Next slide, please. And this is the pivotal trial. Revolution Medicine reported the OS top line a few weeks ago. 500 patients, 300 mg Dara versus investigator's choice of chemo. And its co-primary endpoint is PFS and OS. And it's apparent that RevMed hit both, and very good hazard ratio. We'll see the data tomorrow. One thing I will particularly focus on is on the safety side. In this patient population, when patients are on the drug for longer, so whether we will see higher grade 3 plus GI skin adverse events, what would be the stomatitis, mucositis signal looks like, and also are there other weird things that we don't expect to see pop up in this broader patient population. Next slide, please. Yeah. And there is an interesting study on the resistance mechanism published on Cell, also last week or a few weeks ago by Piero Litto's group from Memorial Sloan Kettering. So he is the top expert that also did a lot of research study on the earlier KRAS G12C inhibitors. So in this study, they sequenced patients of 40 Dara-treated patients of different lineages and find what are the mutations that lead to resistance. And top-line findings is most of these patients are still addicted to KRAS pathway, and the resistance pathway the resistance mutations are the mutations either on KRAS or on the downstream interaction partner of KRAS that make the cancer cells KRAS still active. So one class of mutation is on the Y64CD. So this is where exactly the drug Dara binds and form the tertiary structure with cyclophilin A. So that is a point mutation that lead to the disruption of the tertiary complex formed by the drug KRAS and cyclophilin A. And also there's a second class of mutation is on Y71H. This mutation increased the binding affinity of KRAS with downstream interacting partners, CRAF and BRAF. So basically, because KRAS can bind to RAF more tighter, so it can outcompete over the interaction induced by the drug and cyclophilin A. And finally, another interesting class of mutation is actually on the RAS binding partner, BRAF, CRAF, and SHOC2. So these mutations actually form dimer formation of BRAF and CRAF and structurally make the RAF complex binding titer to KRAS. So instead of direct mutations on KRAS, it can have mutations on the downstream binding partner of KRAS to outcompete the tertiary structure formed by the drug and KRAS and cyclophilin A. So take-home message is most of the mutations are actually still around KRAS, which is great to hear, actually, because that is following the same paradigm as all the EGFR, ALK, and the other receptor tyrosine kinases drugs, that most of the mutations are actually on the oncogene that the drug is trying to target itself, which means that there is an opportunity to develop a second-generation drug still targeting KRAS. And there might be a small fraction of patients that have different mutations, either upstream or downstream, or lineage transformation and other more sophisticated things. But at least from this study, we know that the early signals shows that KRAS molecular glue mutations actually trying to get rid of the inhibition of KRAS, not by directly bypassing KRAS. Next slide, please. Yeah, and this is the overall state of where Revolution Medicine is at. So they are in a good position to pursue multiple clinical studies by themselves. They just raised $2 billion as a secondary, which is a lot of money for a small biotech company. I think they're not small anymore. So they are they're running pivotal trial of Dara in first-line metastatic pancreatic cancer, as well as adjuvant trial. So both of these trials are anticipated to hit because of pancreatic cancer standard care is really poor. And also they are running non-small cell lung cancer trials, as well as KRAS mutation in other solid tumors. And the positioning in non-small cell lung cancer and other cancers might be a little bit different because in non-small cell lung cancer, we have now we have multiple good KRAS G12C inhibitors, and we have more KRAS G12D inhibitors in the landscape coming up. And because of the therapeutic window issue, overall we see that the mutant selective KRAS inhibitors have a better therapeutic window compared with the pan-RAS glue. So we need to see how the data mature in non-small cell lung cancer, where the landscape is busier and what is the competition will be both on efficacy and on safety of the mutant selective inhibitors compared with the pan-RAS molecular glue approach. And also RevMed have their specific G12D, G12C, and G12V mutant selective molecular glues, which in theory should increase therapeutic window. And we need to see more data pan out in the future. Next slide, please. Yeah, and that's something Revolution Medicine presented at AACR earlier this year, which I think is very interesting. It's their second generation molecular glue. It's called RM055. It's still preclinical. And their thesis is they did a different binding pocket and some different chemical tweaking. And instead of making cyclophilin A as a steric hindrance partner to prevent KRAS from binding to RAF and downstream partners, this new molecular glue could actually make cyclophilin A hydrolyze GTP on KRAS better catalytically. You need one copy of cyclophilin A to make 10 copies of on-RAS to off-RAS, which is very interesting. I think mechanistically, still, I don't think the field understands how cyclophilin A could be able to do that. But if that holds true, that would be very interesting that you need less dose of the drug. And also, potentially, you can show a higher therapeutic window as Revolution Medicine showed in preclinical model. And because that's a different binding pocket, this drug could actually work in patients develop resistance to Daraxonrasib, because that's a different, complete different MOA. And that could be the second generation that Revolution Medicine developed, that could either be a solve the resistant patient population or potentially become a better in class RAS molecular glue. It is still very early, but the AACR data and the whole concept is very interesting. And the next slide, please. Yeah, so this is earlier, a few weeks ago, I wrote a summary article in the KRAS space, and kind of on the whole history about how KRAS is being discovered as one of the major oncogene in human cancer, and we have 30, 40 years of failure in targeting KRAS. And finally, we started to see the burst of different classes of KRAS inhibitors, first from the KRAS G12C class, and now we have very exciting data from Revolution Medicine on the RAS on glue, the RAS specific glue, and we started to see more competitors. Erasca is one, and many other earlier RAS on glues in the landscape. And we are only at the beginning of making KRAS druggable. And for pancreatic cancer, it used to be the toughest indication because of KRAS mutation. And now the overall survival rate got doubled. So it's a very exciting time to be in oncology for drugging KRAS.
Yeah.
So
Thanks, Liang, for the sharing. Sure.
Yeah, Liang, that was great. So it's very interesting. So a couple maybe like one or two questions. So you showed last slide another RM255 something, that molecular mechanisms, how they do hydrolysis. I do think that's very complicated and very interesting. But I mean, data of course looks exciting, right? Very different MOAs. But from drug resistance wise, because there is no steric hindrance, do you think this might be more easier to generate resistance potentially?
That's a good question. I think I need to understand the mechanism a little bit better to understand how efficient is the hydrolysis process. And I think in theory it will be less easy to get resistance because as if the molecule is a catalytic molecule, then you don't need the molecular interaction to stay for very long, and you need less copy of cyclophilin A to do the job. If you do steric hindrance, you kind of need one drug to hold KRAS for a longer period of time. So I think if it's catalytic, then the process is more efficient. But I think more about it, thinking out loud, I think it wouldn't necessarily make resistance less likely to occur, because resistance occurs as part of a cancer revolution process. Then if there is a clone that have random mutation and outgrow, it will bypass either steric hindrance mechanism or catalytic mechanism in the equal manner. So I think it's hard to predict. We need to see that in patients, and we will see more data from preclinical models. Actually, now in the KRAS space, we know that a lot of the preclinical models are very translational. So I think we need to see more data mature, both on the mechanistic side and on preclinical resistance models.
Thank you.